Okay, so this is going to be one of our big videos where we're going to talk about the electrical activity in neurons. For this one, I know this is a laundry list of things, so I'm not going to read every point. But the big thing we're going to be going through is basically walking ourselves through the process of how a neuron goes through what's called like an action potential. How, what is resting potential? What is action potential? How does a neuron communicate with the next neuron or a muscle or whatever the effector is? So we'll go through this step by step. We'll try to keep it as clean as possible, but this one is a little bit tricky, so you're probably going to need to spend some time on this one. So let's talk about the membrane first. We got to talk about the membrane and kind of what the potential difference really means. When we say action potential and resting potential, what does that mean? What we're talking about here is a charge, like an electrical charge that is found inside and outside of the membrane. And there's a difference in the charge we find inside versus outside. Cells typically like to be on the more negative side inside with a more positive charge on the outside. So a normal resting cell is going to be about negative 70 millivolts. So this is the potential difference, right? The difference between the inside and the outside of the cell in terms of charge, right? In terms of electrical voltage. So we can actually measure that, right? We take like a voltmeter, we can actually measure that there is a difference, right? If we have like the reference electrode we see here, it's picking up what's on the outside of the cell. Here's our recording electrode that we stick into the cell, and we see that we would have negative 70 millivolts compared to the outside. And this difference in charge is coming from the difference of ions, right? There's a difference in positively charged ions that we find inside and outside of the cell. There is a lot of sodium ions outside of the cell, right? And so a lot of sodium ions means we're going to have more positively charged ions, more positively charged particles. That means our charge is going to lean on the more positive side. On the inside of the cell, we have a small amount of potassium ions. And notice potassium still has a positive charge, but the relative abundance, right, is different. So if we look at the picture here, we see there's only 2 potassiums being shown and 6 sodiums being shown. So there's more positively charged particles, right? There's more positively charged ions outside of the cell compared to inside. And so because of that, the net charge, right, it's more negative inside because we have fewer positive particles. So this is when the cell is at rest. This is what we mean by potential difference, right? And this is how we achieve that, right? Is basically these ions that have a charge, more positive ones on the outside, fewer positive ones on the inside. The sodium and potassium will come into play as we keep talking through this video, so keep that in mind. But the normal distribution of these ions across the membrane of a resting neuron, this is called our resting potential, and so it's negative 70. You're going to have to know these numbers, so keep that in mind. Resting is negative 70 millivolts. Now, when we are a neuron, we are excitable, meaning we can cause a change in that electrical charge. We can change from negative 70 to something more positive, right? And so there's a couple ways that these electrical changes can happen. A graded potential, this is going to allow communication in a short distance in the cell body. And then an action potential, this is going to go along the axon. So graded potential happens in the cell body. Action potential happens along the axon. And there are channels and pumps that are going to move ions back and forth across the membrane that are going to allow for these changes in the electrical charge. So since we're talking about sodium and potassium, we're looking at sodium and potassium channels. Now these channels rely on facilitated diffusion, right? Going from a high to a low concentration, following their concentration gradient to lead to depolarization. We have a couple different types of channels. We have leakage and gated. Leakage or non-gated channels, these are just going to periodically open. There is no stimulus. They just basically think like things just kind of slowly leak out, right? They leak through with a leakage channel. Gated channels have to have some sort of stimulus in order to open. That stimulus can be a change in the local voltage, so voltage gated channel. These can be ligand gated channels, basically a chemical stimulus like a hormone can open the channel, or a mechanical gated channel. This is because of a deformation or a bending, a physical change in shape will open the channel. So we have a couple different versions of the gated channels, right, when we look at our two basic groups. We're going to be looking at some of these as we go through the action potential. So a ligand gated channel, we find this at what's called the neuromuscular junction, or a synapse on the muscle cell, gland, or the postsynaptic neuron. Basically, this is going to be where the nervous system is going to connect with the effector, with whatever it is that's going to be doing the job. They're going to respond to some sort of chemical stimulus or ligand, so like a hormone, or in this case, we're talking about neurotransmitters, right, or NTs. This is going to allow for us to have an action potential happen between the neurons, or from a neuron to a muscle cell or a glandular cell. So if we look at the picture here, right, we have this ligand gated channel. It's closed. In this case, maybe we're talking to a muscle, and this neurotransmitter comes along, in this case acetylcholine. It binds, and now this channel goes from closed to open. Now things can pass through. Leakage channels, again, we find these along the length of the axon, and these just kind of open and close, open and close. There isn't any particular signal that's necessarily triggering this. And so we find more leaky potassium channels compared to leaky sodium channels, which means we're never going to really accumulate a ton of potassium. It's always going to be allowed to leak out, and this helps contribute to that more positive charge outside the neuron and a more negative charge inside the neuron when we're at rest. The voltage gated channels, these we find along the axon as well, and this is what's going to help drive that action potential. This is going to respond to local changes in the membrane potential, so just nearby, right? Not necessarily along the entire axon. And we'll see how this kind of moves in a wave-like pattern in a little bit. But this is going to respond to the depolarization, right? And this is going to promote that depolarization and also promote repolarization. So here, right, we have this -70 charge, right? We see it's closed. And then as the change happens, we go from -70 to -50, we see it opens, and potassium's allowed to float out, right? It's allowed to move out. So we see that as the change in the voltage is what causes this to open in this case. Mechanically gated ones, we find these like on sensory receptors. So like the hair cells in your ear, they physically bend due to the pressure waves, or like tactile receptors, they get deformed when they get pushed on. So these ones, they're physically like being pushed on, they're physically being moved, and so that causes these channels to open, right? So a vibration, a pressure, something like that can open the channel to allow an ion in or out. Now the big ones is the ion pump. This is the sodium potassium pump. We talked about this one back when we talked about active transport, because the sodium potassium pump is like the prime example of an active transport. It's basically the prime example for that. And so the big thing here is we are moving 3 sodiums out and 2 potassiums in against their concentration gradients. This is to help repolarize the cell to get back to our normal resting potential, because if you notice, we're moving 2 positive things in, but we're kicking 3 positive things out. So every time this pump goes, right, we're losing 1 positively charged ion. And that's going to help us reestablish that negative charge, because we're going to be pumping more positive things out compared to the positive things we bring in. So if we kind of walk ourselves through, right, we have our Here is our sodium potassium pump, right? It's open, kind of like cracked open to the inside of the cell, so sodium can kind of notch in there. ATP is used, right? We then add the phosphate, which causes a conformational change. We change, which then pushes the sodium out, but we're stuck in this shape. As long as the phosphate is stuck, we're stuck in this shape, the potassium binds, that's going to kick the phosphate off, which then changes us back to our original shape and allows the potassium to kind of be dumped inside. So that's how we push the 3 sodiums out and bring the 2 potassiums in. So let's take ourselves step by step through what is happening with resting and action potential. So let's talk resting first. Resting, the neuron is more negative on the inside than the outside. So we have a polarization that has occurred in that we are at a negative 70, right? And this is typical. We're going to talk typical here. We're at a negative 70 millivolt charge on the inside. Again, this is because there's a lot of sodium outside and a very little potassium inside to create this change in charges. We're constantly maintaining this polarization by using things like the sodium potassium pumps, as well as allowing the potassium to leak out, right? So we're constantly pushing more sodium out than we're bringing potassium in, plus we're allowing the potassium to kind of escape. Because a lot of the negatively, and you might be wondering, well, why are we just dealing with a bunch of positively charged things? Why are we not bringing in negatively charged ions? It would be so much easier to have a negative charge if we had negative ions. Well, those things really don't have an easy time getting through, and so we don't really see those guys moving very well. So we kind of have to just rely on this difference of more positive versus less positive, right? If you were thinking, going, why? Why is this? That's why. So that's our resting potential. The cells, at this point, the neuron's not doing anything. It's not talking to anyone. It's not doing anything. It's waiting. But then a stimulus comes along, right? And when a stimulus gets perceived, the membrane becomes more permeable to sodium. And remember, the high concentration of sodium is outside. That means sodium is going to want to get in, right? Remember, we have a negative charge on the inside. We don't have a lot of sodium on the inside either. So sodium is going to easily just whoop, come right into the cell. That's going to allow the cell to become more positive. This is what we call depolarization. Right? Depolarization, because we have changed. We're no longer polarized anymore. We have changed from that. This is going to be achieved using voltage-gated sodium channel. And there's 2 outcomes depending on whether or not the stimulus passes a threshold. There is a threshold that has to be met. We can have a graded potential or an action potential. If we're underneath that threshold, we have graded. If we're over the threshold, we get action. So graded potentials, these are relatively weak, right? They're localized depolarization events. It's happening in the cell body or the dendrites, and we don't exceed the threshold. The threshold typically being negative 55 millivolts. So we got a little bit of a change, right? From negative 70 to negative 55, but it hasn't passed the threshold, so we don't actually continue on to an action potential. So nothing happens down the axon. Right? The graded potential kind of stays in the cell body and then kind of dies there, basically. Now, the amplitude, the size of the graded potential depends on the stimulus strength, and it can vary, right? So sometimes they might be just a little blip, right? Like stimulus 1, we saw very, very weak. Stimulus 2, a little stronger. Stimulus 3, a little stronger. But we still did not pass the threshold on any of them, right? But they can be variable, right, in terms of how strong the graded potential is, the size, the amplitude of it. Now, multiple graded potentials follow the rule of summation, and that means we can add them together. And so we add them together to get larger in amplitude by the action of excitatory neurotransmitters. So this is what we would call an excitatory postsynaptic action potential versus an inhibitory. So excitatory neurotransmitters is going to be give us an excitatory postsynaptic potential, versus inhibitory ones are going to be inhibitory synaptic potentials. So the excitatory ones get added together, the inhibitory ones get subtracted from that. And the summation is just the total, right, of this. Like all of them together, integrated together. Do we have more excitatory or do we have more inhibitory? Do we have enough excitatory over the inhibitory to meet the threshold? So if we look here, right, over time, we see here's our baseline basically. We see we have a couple excitatories, we have an inhibitory, we have an excitatory, we have an inhibitory, we have an excitatory, and we pass the threshold. Now we can go through our action potential, right, as we go, when we look at the axon hillock. Right, because that's what matters. That's what's going to decide if we stay here or we pass down to the axon. But this is what we're looking at, summation. Looking at all the signals coming in, some are going to be excitatory, some are going to be inhibitory. Do the excitatories outnumber the inhibitories so much so that we pass the threshold? If we pass the threshold, we then can conduct an action potential. So this is when the graded potential has passed the threshold value, we get depolarization along the entire axon. Now, the weird thing is with an action potential is it's all or nothing. Remember the graded potentials, you could have a little different intensities. We saw the graph where like stimulus 1 was kind of low and stimulus 2 was medium and stimulus 3 was high. An action potential is it happens or it doesn't happen. It's all the same. So we go through that. We then have to, after we go through the action potential, we have to reestablish our negative 70. Right? We reestablish the negative 70 by going through repolarization. So let's go ahead and talk about the action potential step by step. Again, I know this is heavy. We'll be okay. So the stimulus, we get past the threshold, which is about negative 55, right? We pass the threshold, and so now we can open up all these voltage-gated sodium channels and allow the cell to the membrane to really truly depolarize. So we reach the threshold of negative 55, open the sodium channels. Now we're going from negative 55 and we're continuing upward all the way up to like a plus 30, plus 35, right? So all the way up to like a plus 30, because we've allowed so much sodium in, we've allowed so many positively charged sodium ions in. We've gone from a negative 70, we met our negative 55 threshold with the graded potentials, boom, up to positive 30 millivolts. When we reach that, now we allow the voltage-gated potassium channels to open, and potassium leaves the cell. That's going to, and then the sodium channels close, so we're not allowing sodium back in, and we're allowing potassium to leave. This is going to drop the charge back down because we're not allowing any more positively charged things in, and we're pushing a bunch of positively charged things out. Now, we tend to go through what's called hyperpolarization. We go down really low because so much potassium has left, right? So we're hyperpolarized. We're well beyond the negative 70. And then we're going to have to reset. This is where we use our sodium-potassium pumps, right? So that we can push the sodium out and bring potassium back in. Basically, we lost too much potassium. We're like, oh snap, we need to bring that potassium back in. We got to pump. We're going to use a sodium-potassium pump to pump the sodium that we still have inside out and bring the potassium back in. And this brings us back to negative 70. So if we think about it, right, from the start, sodium is outside, potassium is inside. We reach the threshold. We allow sodium in. Now we have sodium and potassium inside. We're positively charged. From there, we need to go back to negative, and in this case, we open potassium channels. So now potassium is outside and sodium is inside. Right? We lock the doors for sodium so we don't allow any more in, but sodium is already trapped inside. We lose too much potassium, We've gone too low. Now we use a sodium-potassium pump to pump sodium back out and bring enough potassium back in. It seems a little crazy, but especially if you draw this out, right, and you assign the numbers and kind of talk about what's happening during each of these changes on this like graph you see here, it isn't as crazy. It's not as hard to digest if you really sit down and talk about what actions are happening and how that's changing the charge. Now there is a refractory period because it takes time to get back to our -70. Because potassium has been pushed out and keeps being pushed out, it's going to take some time for us to close those channels and then use the sodium potassium pumps to get back to -70. So while we're too low, we cannot fire an action potential. So we can only increase in frequency to a certain point, and because of the refractory period. Again, this is when the sodium channels are inactive. There's nothing we can do to overcome this, right, with an absolute refractory period. Relative refractory periods, the potassium channels are still open, and so if you can overcome this, but only if you have like something really strong. Right? But generally speaking, refractory periods, we got to reset ourselves so that we can fire again. And it's that refractory period that's going to help make sure that these action potentials go one way. Right? Because as you're repolarizing, you cannot send a signal. So if we look at how it's moving, right, here's where depolarizing, and it's moving down the channel, but as we're moving down, we're followed behind by repolarization. And that the depolarization can only move this way. We can't skip back over and get to the resting areas down the line. This depolarized region is constantly being followed by a repolarizing region as it travels down the axon. So it's a one-way movement down the axon from the soma, from the cell body down towards the axon terminal. Now, this conduction down the axon, the action potential moving down, can happen 2 different ways depending on if there's a myelin sheath or not. If there is no myelin sheath, we go through continuous conduction, which means we go through every single part of the membrane step by step, right? Whereas if we have a myelin sheath, then we go through saltatory conduction, where we just jump, right? So we bypass, right? We're just going from this empty spot to this empty spot to this empty spot, right? Really fast. So this entire section here, we basically skip right over. That's why it's so much faster. Right? That's why it's so much faster and why myelination is so important, right? And that losing that myelination can cause so many issues for someone. Granted, not every neuron is myelinated. Some of them are purposely not myelinated, and those ones are fine, but for those who have to have a myelin sheath, they need this really quick impulse. They need this increase in speed. So the intensity of the stimulus, basically this is looking at the rate of the action potentials, not the strength. Because remember, unlike the graded potentials, action potentials are all the same, right? Graded potentials, you have variance, right, in strength. Action potentials, it's on or it's off. That's it. So intensity is not from the strength, it's from how many are you getting, how fast, right? And then a stronger stimulus may activate more neurons in a given nerve. This is called recruitment. So we see like the stimulus, right, the strength, boop, on, off. But as the stimulus gets stronger, right, we can recruit more. Right here we're seeing like the action potentials being further apart versus closer together. That's the intensity. Now in terms of how fast this action potential travels along the neuron, that's called the propagation speed, is determined by a couple things. The axon neuron diameter. So we look over here, right, we see like the diameter of the axon, right? That'll increase speed. So the bigger it is, the faster, right? These ones are smaller, so they're slower. And also the amount of myelination. This one has very little has no myelination. This one has very little. They're on the slower end. But that thicker layer of myelination, increased speed. And then temperature, right? As temperature increases, we can also increase speed. So we've traveled down the axon. We get to the end. Now what? Right? Because everything we've talked about so far has all been what's happening inside a single neuron. But how do we then bridge the gap from one neuron to the next? Right? We have to pass over what's called the synapse. The synapse is the junction between what we call excitable cells. So between neurons or between like a neuron and a muscle cell. We have a couple types. We have electrical synapses. We're not worried about that so much. We'll talk about that when we get to cardio. We're not there yet, so don't worry about it. The one we're going to talk about more, though, is the chemical synapse. This is where we have what's called a presynaptic neuron and a postsynaptic neuron. So the neuron that they just sent their action potential down, their axon. The postsynaptic cell is about to receive that signal from that neuron. There is a small gap across there called the synaptic cleft, and this is where we're going to send a chemical called a neurotransmitter out to bridge that gap. So here we have our presynaptic neuron, our postsynaptic neuron, and when they meet up, right, here's the bulb on the terminal, at the axon terminal, and we see all these vesicles with neurotransmitters, and then here's the postsynaptic neuron, one of its dendrites. We see there's that little gap. We got to bridge that gap. So there's a couple things that are going to happen here. Now we're looking at a cholinergic synapse, which means we're looking at acetylcholine being used as our neurotransmitters. And so if we look at what's happening, right, action potential makes its way all the way down. That's then going to depolarize this bulb here, or the synaptic knob. That's going to allow calcium ions in. And then all these vesicles with acetylcholine are then going to be pushed out and release that acetylcholine into the gap. Acetylcholine then travels across the gap, and it's going to bind to a sodium channel receptor. That's going to allow sodium into the postsynaptic neuron. Remember, that's going to then increase the charge inside the postsynaptic neuron from -70 upward. And if it passes -55, we reach our threshold, and now the postsynaptic neuron will be excited and go through its own action potential. Now, we don't want these neurotransmitters hanging out here forever, and so we do have enzymes, in this case acetylcholinesterase, to break down acetylcholine into choline and acetate, which then get brought back in and rebuilt, and then we build those vesicles again. Right, so we bring back the choline, the acetate's gone, we use acetyl-CoA to put the acetate onto the choline, bam, back together. So we're recycling like half so much of the neurotransmitter. But that's why we can send a signal out, right, and then break that signal down, reuse as much as we can, set ourselves up again. So postsynaptic potentials, this is where the postsynaptic neuron, it's going to get a bunch of different signals, different types, all at the same time. We mentioned some of these already in summation. We're going to talk about it again. But the excitatory postsynaptic potentials, the EPSPs, right, these ones are going to push for depolarization, which means they're pushing for an action potential. Inhibitory postsynaptic potentials are hyperpolarizing. So they're going to bring the charge even lower, right? They're fighting to bring the charge even lower than it is, which goes against sending an action potential. So it blocks an action potential. So that's why we call them excitatory and inhibitory. Now, summation is looking at all the signals that are being received, right? So if we look at this picture here, we see that we have these guys here in blue and these ones here in red. We have to look at and take into account all of them. What's the overall signal being sent? And that's summation. So here we start at our baseline at -70. Here we're counting 2 of those excitatory ones, one inhibitory, one excitatory, one inhibitory, one excitatory. It puts us up high enough and we go through our action potential because we passed the threshold. So we're just kind of putting them all together. If there's more excitatory than there are inhibitory, and the excitatory ones push us up over the threshold, action potential is sent. If the inhibitories outnumber the excitatories, then we do nothing. If the inhibitories outnumber if the excitatories outnumber the inhibitories but not enough to pass the threshold, we still don't do anything. Right? It all comes down to, can the excitatories get us up over the threshold even when there's some inhibitory ones there? Now this can be spatial, this can be temporal, right? So here, right, spatial, we see that we release neurotransmitters at the same time, right? So if a bunch of presynaptic ends release their neurotransmitters at the same time, the combined graded potentials can be added or subtracted. So here we get it from neuron 1 only, and it takes us back down. We didn't meet the threshold. Over here, right, we have the release from 1 and 2, right? Or we have the successive release of neurotransmitters from one neuron, but it's releasing it, they're still there, we release more, right? So basically spatial is you're getting it from more than one neuron. Temporal is one neuron is releasing neurotransmitters and then releasing them again, right? So it's one neuron is kind of working as two in the form of temporal summation. Okay, so now look, I know this one was heavy. Take some time to sit with this one though, because this is a major topic. This is going to be a major thing that we're going to be looking at in this unit, right? We're going to talk about all the other things, but this is the meat and potatoes. This is the baseline for a lot of other things that we're going to talk about. This is what's the generic physiology of neurons. And so we have to understand this one to get through this course, and at least to get through this unit. Take the time with this one. Please take the time with this particular video.